Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about the cell cycle. The cell cycle consists of M phase and interphase. M phase consists of mitosis and cytokinesis. Interphase consists of G1, S, and G2. Interphase is about 90% of the cell cycle. Like I stated, there's three subphases within interphase. G1 phase is the first gap phase, and it has variable lengths. The next subphase is S phase. It is the synthesis phase, then followed by the G2 phase. In G1, the cell will grow. All of the cytoplasmic organelles are going to replicate, get bigger, and get longer. During S phase, the nuclear content will replicate. Anything that has not completely and gotten bigger, longer, or doubled will complete their synthesis, their enlargement in G2. The cell grows during all three of these phases. This is a typical human cell. It takes about 24 hours to replicate. The cell spends about 5 to 6 hours in G1 phase. This is where the organelles are going to replicate. If you have 5 mitochondria, now you're going to create 5 more. If you have 5 millimeters of Golgi apparatus, you're now going to create 5 more. Then we have synthesis, S phase. The DNA will replicate during this phase. This takes about 10 to 12 hours. Then finally we have G2. This takes about 6 hours also. The shortest phase is M phase. This takes about 1 hour. Most cells in the human body are in a non-dividing state called G sub 0. Examples of such cells would include muscle cells and nerve cells. Some cells, such as the liver, is also in G sub 0, but it can be called out of G sub 0 if growth hormones are secreted or injected. So yes, in the right situation, liver cells or hepatocytes can be called out of retirement. They can be called out of G sub 0. So the removal of a lobe of the liver cannot be regenerated. But if a few cells are removed, it can be regenerated. The issue with regeneration is if you destroy too much of the scaffold, then the replication, the resurrection of these hepatocytes cannot occur. So it depends on how much of the liver has been damaged. This slide shows that even some muscle cells can be called out of retirement if they are suspended in G sub 0. In certain cases, anabolic steroids can call these cells out of G sub 0 phase. One such cell that always has the potential to divide is the satellite cell. The satellite cells are cells that are embedded within the muscle tissue itself. It is a type of muscle cell that retains the ability to keep dividing. Cell division is for body growth and tissue repair. Mitosis is a process that will divide the nuclear content. Cytokinesis is a process that will divide the cytoplasmic material. We're going to now look at a muscle tissue and how to grow it. There's two ways of growing muscle tissue. One is by adding muscle fibers within the muscle cell. This is called hypertrophy. The other is adding new cells. This is called hyperplasia. Hypertrophy of muscle occurs when new muscle fibers are laid down adjacent to damaged muscle fibers, or by enlarging the diameter of the actual fiber itself. The fiber is called the myofibril, and it is composed of actin and myosin. Later on, when you take anatomy and physiology, you'll learn about the sliding filament theory, which utilizes those two cytoskeletal proteins. In the image to the left, we see four equal sized cells. They're shaped as a square. And if you enlarge the overall diameter of the fibers by adding more actin and myosin, the cell will enlarge. This is how most of us gain muscle mass, is through cell hypertrophy. Another way to gain muscle mass is by hyperplasia. Hyperplasia is going to add a new muscle cell itself. So if we look at the image again, those four cells of equal size that are squares, we're going to add two more squares. This can be done by taking anabolic steroids or growth hormones. This is a concept map to summarize all of this. Some cells, like the pancreas, divide very infrequently, whereas some cells, like the skin cells, divide always and all the time, very frequently. And thirdly, some cells don't divide at all, like muscle cells, nerve cells, and liver cells. When is mitosis needed? During reproduction. Once you have created the sperm and created the egg, those two cells must unite. Once those cells unite, you have created a zygote. Now, the zygote must go through cellular division to make 2 cells, then 4 cells, then 8, then 16. So this is reproduction on the cellular level. Growth and development. As your liver gets larger, cells will divide. As your muscle gets larger, the muscle cells will divide. This is growth and development. And finally, tissue repair. If you get shot in the liver, the hepatocyte will repair itself by creating a new cell. So this is trying to show you how important mitosis is. So we have covered interphase. Now we're going to talk about M phase. M phase consists of mitosis and cytokinesis. M phase has mitosis, and mitosis has 4 stages that we're going to go through extensively. These 4 stages or phases are called PMAT: prophase, metaphase, anaphase, telophase. During lab, you're going to look at all four of these phases. You will locate these phases in a specific region of plants. The plant that we will use is an onion. So onion root tip or onion shoot will contain a region called the apical meristem. This is going to be where the cells are dividing. During interphase, protein synthesis will occur. You're going to make all of the necessary enzymes to sustain life for the cell. DNA synthesis will occur during interphase. And replication of cytoplasmic organelles will also occur in interphase. The nucleolus is visible along with uncondensed chromatin. So when you look under the microscope, you're looking for a dark staining nucleolus. Everything else around that will be fine granules of chromatin. This is still genetic information. It's just not super coded yet. So remember, DNA, when it is going through cellular division, we call it heterochromatin. Heterochromatin is wound up in reference to euchromatin, which is not so wound up. During early and late prophase, the following will occur. The centrosomes will move apart. These centrosomes were replicated during interphase. The centrosome is going to produce spindle fibers that are going to move the chromosomes. But the chromosomes cannot be moved yet until the spindle fiber is made. But then the spindle fiber cannot attach to the chromosome until the nuclear membrane disappears. So the nucleolus will also disappear during prophase. And the chromatin that was condensed is now going to condense even more. Therefore, you'll be able to finally see the actual chromosomes. So visually, under the microscope, you can only see the chromosomes. You can see that the nucleoli is gone. So look for those two defining characteristics for prophase. Some books break prophase up into early, middle, and late. So during early prophase, animal cells will contain a pair of centrioles within the centrosome. What creates the spindle fiber is the centrosome. Scientists have not proven the purpose of the centrioles yet, but they do know that plants do not contain the centrioles. What they do know is that the centrosome is going to make this microtubule that is made of a protein called tubulin. We commonly call this entire filament the mitotic spindle or spindle fibers. Later on, during late prophase, the chromosomes are now attached to those spindle fibers. The chromosomes are replicated during interphase, and now they are ready to move. Some books call late prophase prometa. That replicated chromosome consists of two sister chromatids. Each sister chromatid is a DNA molecule. Each DNA molecule has a centromere. The centromere is going to split. The DNA molecules are going to pull apart. The sister chromatids are going to pull apart. The chromosome is going to pull apart. So the chromosome has been replicated during S phase of interphase. You have to call it a replicated chromosome, which is only one chromosome, until the centromeres split that hold the two sister chromatids together. So the replicated chromosome is two DNA molecules held by three protein complexes called the centromere. So let's talk about the centromere. The centromere is composed of a repetitive sequence of DNA and three associated proteins, protein 1, 2, and 3. Protein 1 is going to cause binding of centromeric DNA to centromeric DNA. So remember, in interphase, a chromosome, which is composed of one DNA molecule, is going to replicate. When it replicates, everything is replicated, even the centromeric DNA. Protein 1 is going to bind to the centromeric DNA and cause those two identical sequences from identical chromosomes to stick together. Therefore, we only call it one replicated chromosome. Protein 2 is going to cause further condensing of DNA, resulting in a narrow appearance. So everybody draws the chromosome as an X. It has a skinny waist. What narrows the waist of this chromosome is protein 2. Protein 2 is like a garter belt or a weightlifting belt. It is used to squeeze the inner portion of an individual. Protein 2 is going to squeeze the inner portion of the chromosome. So now we have protein 1 and protein 2. The third protein is going to form the kinetochore. This is going to be the attachment, the hole that the spindle fiber will attach to. So again, the centromere is composed of protein 1, protein 2, and protein 3. This slide shows that the microtubule, or the spindle fiber, is attached to that hole. That hole is the kinetochore. It is part of the centromere. What makes up the microtubules? Tubulin. Tubulin is the protein that makes up this microtubule. The next phase is metaphase. So we have now created the centromere. We have now created the spindle fibers that are made of tubulin. We grew them towards the centromere. It has now attached to the kinetochore of the centromere, which is protein 3. Now we're going to move the chromosome. We're going to play tug of war. The centrosome is producing or removing the spindle fiber. So it's going to push and pull the chromosome until the chromosomes line up on an imaginary plane. The portion of the chromosome that lines up is the centromere. They will all be located on what is called the metaphase plate. This image is a stained cell with the green elements or spindle fibers that have been stained with the green stain. The chromosomes are staining blue-purple. They are all lined up on an imaginary plane. The next phase is anaphase. During anaphase, centromeres of a chromosome will split. Remember, the chromosome is nothing but a replicated chromosome that has two identical DNA molecules. Those identical DNA molecules are called sister chromatids. During anaphase, the centrosomes are going to pull and split the centromere apart, therefore liberating the sister chromatids. Now you have to call the sister chromatids chromosomes. What pulls? A motor protein in the kinetochore is going to pull the chromosomes toward their respective poles. The poles are where the centrosomes are located. This slide shows you the DNA molecules moving back to their respective poles. And this image is a north pole and a south pole. The next phase is telophase. During telophase, new sets of chromosomes will unravel back into chromatin. A new nuclear membrane is formed from the rough endoplasmic reticulum. The nucleoli will reappear. Generally, cytokinesis completes cell division. So what you'll be able to see under the microscope is a cell plate to illustrate cytokinesis is starting to occur. And you will be able to see chromatin. No longer will you see the chromosomes themselves. So we are done with PMAT, prophase, metaphase, anaphase, and telophase. Now we're going to look at the division of the cytoplasm. This process is called cytokinesis. We will look at cytokinesis in animals and in plants. Cytokinesis in animals occur when the cytoplasm is pinched into two parts after mitosis ends. This begins during late anaphase when a cleavage furrow is formed by contractile rings. So think of that weight belt being placed around a circle, and you tighten the weight belt. The weight belt is the contractile rings that are made of a second cytoskeletal protein called microfilaments. This is going to squeeze the cytoplasm to the right and to the left. This is observed in animal cells. So in lab, you will have not just the onion root tip, you will also have animal fish slides. On the animal cell, you'll see the cleavage furrow. So in this slide, we're looking at a cleavage furrow. This was created by those microfilaments. This is cytokinesis in plants. In plants like the onion root tip you'll look at in lab, you'll see what is called a cell plate. This cell plate is formed by Golgi vesicles during late anaphase. So the Golgi vesicles are laying down side by side to produce what will eventually be the cell plate. In the end, the cell plate will fuse with both ends to create two new daughter cells. This slide shows all of the stages we talked about. You should be able to identify each one and call them out by name. These are the names of all of those stages. This is an animal cell that is going through mitosis.